Hi, I'm the Senior Director of Population Health at Olink Proteomics, and it's my distinct pleasure to be here to share ways that you can access proteomics, broad-scale proteomics, across the proteome, in plasma samples, and anything you can make liquid. So CSF, even isolating exosomes from within plasma. You can analyze the proteomic profile to better understand what these exosomes are carrying within the blood, and in many cases we think this is how signals are going from the blood to the brain, past that blood-brain barrier. So being able to look across the proteome, look at proteomic signals broadly, is an exciting advance in complementarity to mass spectrometry. This is now accessible on the T7, so I'm excited to share a little bit of data and a little bit of application with you today. So the proximity extension assay, and this is a short video that will loop, so if you miss the first bit there, you'll see it come right back around. The way that our assay works and the way that it's really, I think it's differentiated, is we have two antibodies that find each protein, so we're not pushing a large volume of sample through a column in a mass spec instrument in this assay. We call it an affinity assay. Those two antibodies, once they find the protein that they're designed to find, they have these single-stranded oligos that can then hybridize. Once they hybridize, you can extend them and amplify them just like any library prep. Then it becomes a counting exercise to count how many of these fragments represent the original amount of protein in the original sample. So you're changing your complex protein signal to what I think is a simple DNA signal. So the T7 now it's got this beautiful capacity to be able to read out over 5,400 proteins at a time. This is our maximum throughput of proteins, maximum coverage of the proteome. We're targeting full-length proteins here, and I think you'd argue that there might be 20,000 proteins in the body before we look at proteoforms. I think we're doing a great job of covering the ones that we expect to be leaking into the plasma, as well as some discovery proteins, and I can always share the profile of the proteomics that we target, or you can download it from our website. So the team that has been the first user of the proteomics on the T7 is at Sequanta. This is the team there. Very impressed with their bringing proteomics to the Asia market. And here are the data. So on the left, we're showing the correlation across all blocks and plates. When I say blocks, the way that we manage dynamic range in this proteomic world is that we're doing dilution blocks of the samples themselves, and the blocks of proteins are the ones that are high abundant in one block, and then we have many low abundant blocks in order to manage and capture the full spectra of the abundance we expect in these samples. And then informatically, we bring them back up to comparison levels to be able to then look often from cases to controls, identifying biomarkers. Now, I just wanted to include a little bit of biology about these. So we were able to stratify the males and the females in this and look at proteins that were expected to be different between genders. And we're definitely seeing the biology we expect. Not quite a one correlation, because that would be suspicious, but the correlation on the left is 0.994. I have to make that joke because you made that joke. And this is the full product portfolio of sequencing instruments that Complete Genomics offers. Right now, we're offering on the T7. That's what we've validated on. Look to the future. We'll see. You never know. And so a little bit more about proteomics and how people are using this capability, particularly in the context of a world where the gold standard really has been mass spectrometry. Where we provide a lot of value is in this range, in this lower right quadrant. So just to orient you on this graph, along the x-axis, this is just sorted proteins from the highest abundance in the upper left to the lowest abundance in the lower right, the abundance expectations. This is on across a dynamic range, but the abundance expectations is on the right, the scale. And we're really targeting and focusing, you can see the density of these dots on that low abundant plasma proteome. Now this is where the cytokines, the chemokines are, where mass spec does a fantastic job, and we've already got lots of clinical tools, is in that upper left quadrant. We have certainly some high abundant proteins, but we're here to shine a light on an area of the proteome that just hasn't been easily seen using other approaches. And we've got over 1,600 peer-reviewed publications by customers to reflect some of the biology that this is enabling. And many of the large population biobanks are leveraging these data. I'll talk a little bit about the UK Biobank in this upper right, and then what's called the SCALLOP Consortium, which is a consortium of consortia, and that's off to the left. So the UK Biobank has generated data for over 54,000 samples. These are mostly baseline samples from the UK Biobank. This is the largest proteogenomics project that has ever been done, and it's called the UK Biobank Pharma Proteomics Project. Now they're just scratching the surface of what they're able to do with these data, and they're publicly available on the DNA Nexus Research Analysis platform. So I won't go into detail of what all these pharma are doing with these data other than mapping these relationships between genetics and protein levels. 3 papers came out in October's, a single issue of Nature in October of 2023 that talks about this atlas, this mapping. But they didn't take it beyond that in those papers because they're working diligently behind the scenes to really map therapeutic targets. So I'm going to talk about the SCALLOP study, which is really a powerful precursor to what the UK Biobank is doing, but on, like I said, a larger scale. So here's the flagship paper of the SCALLOP study. This is 90 proteins in this study. It was published in 2020, and over 30,000 individuals. Now these were individual cohort studies that were then brought together, harmonized and meta-analyzed together. And what they're doing with these data, and this reminds me a lot of those GWAS chromosome maps, these are chromosomes 1 to 22, is they're mapping the relationships where they're seeing significant associations between regions in the genome and protein levels. Sometimes these are cis associations, so sometimes these are ones between a cognate gene and the protein that it codes for. But sometimes these are trans associations that are representing interactions between proteins within the genome, that are coded within the genome. And so, like I said, just scratching the surface of being able to map these relationships. With this publication, what the team did was they mapped these associations, then they overlaid the clinical data from the UK Biobank, and then they were able to do this Mendelian randomization, whereby they were able to resample from these 3 pots of data to then postulate which of those associations between genetics, proteins, and disease were unlikely to happen by chance alone. They were then left with a subset of pathways and diseases that appear causal. So this is much more valuable than 450 pQTLs, or relationships between genetics and proteins. Pharma would much rather have a subset that are the ones that appear causal for them to then investigate than a long list of associations of proteins with disease. So being able to tease out this causality is incredibly important for improving our ability to build therapeutics. From the 25 out of this set that appeared causal, there were 14 that were corroborated or recapitulating known therapeutic targets. So good evidence that we're on the right track with this data approach. Some of those are shown in the blue circle. There were 11 novel targets out of these 25. Those are the ones shown in the green circle. This was funded, much of it was funded by Pfizer, and Pfizer has stated publicly that at least 2 programs came out of this original data from SCALLOP. I don't know that they're on this slide. We won't know until they come out with their programs. but exciting advances within pharma. And then this purple square I'm particularly excited about because these represented relationships between pathways and diseases where we already have therapies to target those pathways, but not in those diseases. So these represent potential repurposing opportunities that pharma can explore. So I'm going to dig into just chromosome 1 and talk about each of these 3 categories. So corroborating known therapeutic targets, new novel targets, and repurposing, potential repurposing opportunities. And I just picked chromosome 1 because it's nice and big and has all 3 of these examples on it. So a validated target on chromosome 1 is for IL-6R. And indeed, tocilizumab, which is a therapy from Genentech, is targeting this pathway in rheumatoid arthritis, an approved therapy. A novel finding on this chromosome was for F3, which codes for a protein TF, and this was associated with HbA1c levels, so might be relevant for metabolic disease, has to be explored. And then 3 repurposing opportunities map to this chromosome. I'll just focus on the one in the middle, that's that IL-6R. So the way this works, you see how it says asthma, eczema with a little asterisk, and then AFib. So the idea here may not be a good idea, but certainly it can be explored by pharma and they'll prioritize it. But the idea here would be that tocilizumab, which targets this IL-6R pathway, might be repurposed here in asthma or atrial fibrillation. The asterisk is about directionality and suggests that eczema could be a side effect of targeting this pathway. And so I dug into the label on tocilizumab, and indeed eczema is a side effect, a possible side effect that showed up in the clinical trial. So great, exciting, I think, and some validation of these results. You don't have to have 54,000 samples to make use of the data that are provided through proteomics. So many folks are using our qPCR platform to build out an understanding of relationships between proteins and disease in biomarker studies that don't include genetics. Here are some that are facilitating our insights within cancer, and I've categorized them. These are just publications that I picked out of our 1,600 database. These are ones that are relevant in early detection of cancer, disease progression, tracking cancer as it's progressing. This is important in tracking people who've been in remission. And then treatment response, being able to stratify patients to understand what treatments might work for them. So that's in cardiovascular, or sorry, in cancer. And here are similar publications or publications relevant to cardiovascular disease. I will say in cardiovascular disease, this is part of our blood system. We really laid the groundwork of our capability to show insights in this area, whereas cancer publications are just starting to gain momentum. We just didn't have as many of those early on in our cycle of development as a company. And I think this is because some of those cancer markers really, we didn't expect necessarily to show up in blood. It's pretty delightful to see that they actually are providing a lot of value, and I think will layer onto our understanding of early detection approaches with cell-free and methylation, cell-free DNA and methylation, in order to mediate some of the false positives we're seeing in some of those approaches. So I mentioned our qPCR readout, so I'll just put in context that our broad 5,400 proteomic tool called the Explore platform that's available on the T7 is what folks are using to really narrow down sets of markers to then build out a tool that can then be suitable to take to the clinic. Our progression of this in our product names is from the Explore to the Target for validation. These are 92-plex and 45-plex proteomic panels. And then eventually customers, once they've figured out what the subset of proteins are that are relevant to an application or a disease, they can build a flex or a focus panel. So this is sort of our continuum. And so next, and just to finish up, I want to just give you some flavor for some of the flex and focus panels that have been developed and are going through clinical validation. Here's 2 of them. So one is built by a company called Octave Biosciences in the Bay Area, and these are 2 of a dozen different examples of customers that are building these potential clinical insights. The Octave Biosciences targets multiple sclerosis. The one on the right is built by the Crohn's and Colitis Foundation from their discovery efforts, and this is relevant in IBD, inflammatory bowel disease. Both of these are pretty challenging diseases that can be diagnosed quite early in life, meaning a long time period of moving from one therapy to another therapy. On the one on the left is this purpose is really for disease progression and understanding when it's time to move from one therapy to another. And it's part of a dashboard, this set of proteins that gets run over multiple times over the course of someone's monitoring of their disease. It's part of a clinical factors and mobility factors in this dashboard that physicians can use that Octave provides. On the right, the purpose for this IBD is it was developed in a pediatric cohort. I like these 2 because they're so different. In this pediatric cohort, they started out with samples that were collected at diagnosis in these kids. And what they were able to follow those kids in their progression of disease to develop a score to understand the likelihood of complications to develop in IBD so that they could insert a pause before taking out a kid's bowel if they scored indicating that they were unlikely to develop complications. So pretty phenomenal team there at Crohn's and Colitis as well in building out this capability. Both of these I like as examples because they both have a path to the clinic. On the left, Octave just closed its latest series, actually it was about 6 months ago, that they closed their series of funding for taking this to the clinic. They're the furthest along, so my next slide is about that clinical validation that was done by an independent party. The one on the right, Crohn's and Colitis is a nonprofit foundation, so they launched an IBD Ventures arm, and they've partnered with a diagnostic company in order to take this to the clinic. I like it when there's actually a path, right? Because it does take money to get through that clinical utility. And then this came out in July of last year. This was done through the Brigham and Women's team that independently validated the Octave Biosciences clinical application. So I expect that to show up as an LDT very soon. So I hope that I've been able to give you a flavor of how our technology is being leveraged for building a systematic approach to identifying therapeutic targets. Biomarker discovery is, something we think of when we think about proteomic technologies. We're able to help facilitate biomarker discovery, but also with mechanistic insight. So I think it's a 3-for-1, which a lot of folks are really getting excited about. And so I'm just pleased that we can now make this accessible for those that are using the Complete Genomics instruments. So thank you for your time. Happy to take some questions.